So our next presenting company is ReNeuron, and this is a cell therapy company based in Wales in the UK, if I'm not mistaken, and traded on the AIM exchange in London under the ticker RENE. Here to present for the company is CEO Olav Hellebo. Welcome, Olav. Thanks for being with us.
Thank you, Ted. And I thought I was your favorite person. So disappointed now, coming in second. And I always enjoy seeing Linda talk about exosomes, so because we have a shared passion there. ReNeuron, we are based in Wales. That's right. We moved actually from the London area to Wales about a year ago to great new facilities down there. I have a few slides here just to kind of get us warmed up. We are looking into treating patients who've had a stroke. We are not in acute stroke. So acute stroke is an area where a lot of people have tried to develop new drugs and failed. TPA is available from Genentech. Has to be used within the first 3 or 4 hours. Athersys hopefully will come with the next one within the first 36 hours. But after that, a stroke patient is terribly sick, gets a lot better within hours, days, weeks. After a couple of months, they're quite stable, but they're not 100% well. Typically, they have disabilities that really limit them what they can do. And our goal is to kickstart a new cycle of recovery in those patients. That is very much pioneering. No one's done that before. There are no therapies available to help patients who've had a stroke. Obviously, there's a huge potential market, stroke being the #1 cause of disability in adults. We have shown our phase 2 results a few months ago. Our phase 2 was a single-arm study in 21 patients. We delivered 20 million neural stem cells through neurosurgery into the brain of these patients to help then kickstart recovery. We saw results of improvement in four different efficacy measures that we used. 15 out of the 21 had improvements across these measures. The one measure that we put in there and we didn't really expect to see much was modified ranking scale. Modified ranking scale is what Genentech got TPA approved on. It is what FDA likes to see, and it's the one that's killed a lot of drugs because it's very difficult. It is a very chunky scale. It goes from 0 to 6. 0, you have no symptoms, and 6, you're dead, which is a tough scale. So 5, you're bedbound, 4, you're wheelchair bound, etc. So moving one step on this scale is hugely meaningful. And we were able to do that in 1/3 of the patients. So that really sets us up now to go into a phase 3 study. And we're actually talking to the FDA tomorrow. That's our end of phase 2 meeting tomorrow. So the plan is obviously then to kick off with the phase 3 study later this year, mainly in the US. We'll probably have some centers in the UK as well. And we'll keep your eyes open. You will see some news coming very soon on that. The second product we have are human retinal progenitor cells. These are cells that have application across different retinal diseases. Our first disease that we've started is retinitis pigmentosa. We actually just announced that we are also going into a second disease called cone road dystrophy, CRD, which is kind of the opposite of RP in a way. They lose their cones first rather than rods as you do for RP. We have orphan drug designation and fast track by the FDA for this program. And we have a phase 1 study ongoing right now here in Boston at Mass Eye and Ear. And Dr. Eric Pierce is the investigator for that study. We finished the first 2 dosing groups in phase 1. And there's one more dose group to go, which is a 1 million cell level, which most people expect to be the right dose level. We have just filed our cryopreserved formulation. So we're waiting with the last dose to use the cryo, waiting for that approval so we can use the cryo for the last dose cohort so we get some safety data on that. And as soon as that's done, we will do a phase 2. We just announced that we have expanded that from 6 patients that was originally designed to 20 patients, so we can have more solid data to make a decision going forward on that. So in summary, I'll leave the summary up here while Ted interrogates me, I think. Thank you.
Great, Olav, thank you for that brief overview. And maybe I can take half a step back and get a little bit more into the detail about these Ctx cells. What are they? How are they derived? How are they delivered in the stroke setting?
So they're fetally derived neural cortex cells. We bought them from a cell bank or tissue bank in California about a decade ago. We use a gene technology called c-MYC to immortalize those cells. So what it allows us to go through many more passages than was normal, which means that we never ever need to go back and rederive. So it's an everlasting supply. We say every cell is a clone. So this is allogeneic therapy. They are cryopreserved, which makes it possible to do a multi-center clinical trial. Back in phase 1, we had 14-hour shelf life. That was difficult. We still did it. Now we got 6 months shelf life on the cryo, and we can just expand that. We tend to use them up fast, but so that's the cells. The cryopreserved makes it possible to deliver straight to the hospital. All they have to do is to take the vial out of the container and let it just warm up in ambient temperature and then inject it. It's done via neurosurgery using a stereotactic frame. Takes 2, 3, 4 hours. Most of that time is setting up the frame. The injection itself is quite easy.
And are the injections really in the infarct area? And what kind of doses are you administering?
We are using the top dose from our phase 1, which is 20 million cells. That showed good efficacy in phase, or signs of efficacy, let's call it, in phase 1. And in phase 2, we had that confirmed, so 20 million is our dose. They are always put in the same area in the basal ganglia, which is a great source of blood supply for the rest of the brain. We do not put the cells into the infarct area. In animal studies, we can see that the cells don't do well. They need to be in healthy tissue to do well. So that's where they go.
So by inserting them there, they actually port sort of to the site of injury themselves? Is that the right way to think of it? Or do they stay local and really more excrete paracrine factors that result in the healing?
Yeah, no, it is paracrine factors, and just like with Capricorn, when we were looking for mechanism of action of our stem cells, we think exosomes are probably a big part of that. And yeah, it's very much about kickstarting repair. The brain is amazing, and the brain is really good at adapting to a damage and working around it, and that's what happens in acute stroke. When everything is stabilized and the patient is not improving anymore, here's the opportunity to kickstart it one more time, to get repair going on for a few months. You probably will end up with a new plateau, would just be a better one.
Excellent. So I want to ask about, and again, I guess we'll know more on Friday after, or I guess on Friday afternoon after you've met with the FDA, but what do you envision as the design of the phase 3 study? How large would it have to be? And would you go after modified Rankin as the primary, or is the FDA more interested in composite stroke endpoints at this point?
The plan for the phase 2 is 220 patients. 1-to-1 randomization. You might wonder how we do placebo control for brain surgery, and it is called placebo surgery. So the surgeon will not be blinded. I think patients will be happy to hear. But the patient will be blinded, and the assessor will be blinded. Some surgeons, you wonder. The patients will be sedated. The surgeon will actually drill a hole in their head, but it will actually not penetrate the dura, so there is no infection risk to the patient, so nothing is actually injected. But the patient will think that he or she had surgery, and they'll maybe try harder, which is the whole placebo effect, right? So, we expect that there will be a placebo effect in our study, and we've taken that into account into our design of the study. We're aiming for a 20% point difference between placebo and active. In terms of endpoints, we'll know tomorrow. I know FDA will love modified ranking, so I think we're pushing an open door on that one. But activities of daily living in general is what both FDA and EMA is looking for, rather than some made-up kind of scales.
Great. I want to see if there's any questions before we move to the RPE program. So I've really always kind of thought of RPE cells as sort of nurse cells to the cones and rods. So maybe you can tell us a little bit more about kind of how these cells are working in the eye, and when can we get data from the phase 1/2 study?
So these are actually retinal progenitor cells, and again, pioneering medicine here. What we hope that they will do in humans, that we've seen signs of that in animal studies, that they will make new photoreceptors. So you're born with all the retinal cells you'll ever have. Your eye is not going to make any new ones. So when they're gone, they're gone. And it couldn't be a better place for actually cell replacement therapy.
Interesting. That's cool. Are you able to actually monitor that and actually see an increase in number of?
Well, I mean, in animal studies, what we can see is we can see connectivity through the ganglion layer into the optical nerve. We don't know if that means that actually there's a function that has been restored. You just can't do that in those kind of models. We'll have to wait to ask the humans to see how they do on that.
And you mentioned exosomes, which is one of my favorite areas in biology right now. So do you have any comments on what your early efforts are in that nascent field?
So exosomes is obviously very, very interesting. We are pursuing exosomes in two different ways. One is to use them as a therapeutic agent, like we heard from Linda just before. And the second one is actually to use them as a delivery vehicle for gene therapy. We have spent the last year working very hard on CMC. It is just extremely important that every batch is the same, and that's not easy to do. Obviously, the starting point is a GMP production batch for our Ctx neural cells. So, we're starting at a good quality point, but it's very important when you take them out of the process, you take them from the media, and if you take them from an early stage or a late stage, they're completely different. So we have characterized two different exosomes. One is early in the process, the other one is later, when we have more proliferation that has happened. And actually, one is anti-inflammatory, but the other one is pro-inflammatory. So they're different. We got a £2.1 million grant to work on this together with Cell Therapy Catapult and NKI in the Netherlands. So we have now what we think is a stable product, which is well characterized, that we can produce in high volumes.
Very interesting. And just with the last few seconds that we have left, how much cash do you guys currently have, and how long will that fund the company?
We have $77 million in cash at the last accounts, which is a lot of money, but we also have a lot of ambitious programs. So, running our ambitious program, that should take us to the end of 2018.
Great. Excellent. Olav, thank you very much for being with us today. Thanks, everybody.
Thank you.